Tetanus is a serious disease caused by infection with the bacterium Clostridium tetani. Clostridium tetani is found throughout the environment, especially in soil. The bacterium produces spores which give it its characteristic tennis racket appearance and make it very resistant to heat and antiseptics. The bacterium usually enters the body through breaks in the skin. When the bacteria reaches a suitable environment, the spores germinate and begin to divide. They then produce a toxin called tetanospasmin, which disrupts normal neuron function. Symptoms of generalized tetanus include lockjaw or jaw stiffness, headache, muscle spasms, difficulty swallowing, and irregular blood pressure. In extreme cases, the spasms can lead to bone fractures and aspiration pneumonia, and prolonged hospitalization from the illness can lead to secondary infections.
To understand how the vaccine functions, let's start with how the vaccine is made. Clostridium tetani are cultured in a lab under favorable conditions so that they produce the tetanus toxin. The toxins are then extracted and mixed with formaldehyde. The acidic mixture causes the tetanus toxins to denature and unfold to a point where they cannot naturally fold back. This prevents the toxins from binding to the peripheral neurons when injected into the patient. Because the toxins cannot enter and essentially hide in the neural cells, they now float freely in the body fluids, where eventually a B cell can pick up a few and present them via MHC class II to a helper T cell. Once the T cell recognizes the antigen as foreign, it will command the B cell to produce the appropriate antibodies to combat the toxins. The antibodies neutralize active tetanus toxins by binding to them, essentially preventing the toxins from entering the neurons similarly to the formaldehyde treatment. And thus the neurons, and more importantly, the body, is saved from another tragic event. Tetanus toxins bind to peripheral neuron receptors and are endocytosed into the neurons. Then they trick the neurons via disruption of the cell's internal pH regulation into passing the vesicles containing toxin toward the motor cortex of the central nervous system. There, the toxins inhibit the release of gamma-aminobutyric acid, aka GABA, and glycine, which act as neurotransmitters to inhibit motor neurons to help control motion. Additionally, they degrade synaptobrevin, an excitatory neurotransmitter. As a result, the motor neurons become hypersensitive to stimuli, and even the slightest stimulation can launch the body into a volley of severe spasms.